I will be covering Principles of Inheritance. Briefly, I will run through how to access revision notes and test in classix.com. Open classix.com page, click on Practice. So you will see a lot of packages. Select NEET Premium because we are doing the NEET course here. And here you find chapter-wise tests in physics, chemistry, biology, and NEET full tests. Now here you see a lot of colored bubbles. Now each bubble is a test. A green bubble indicates that you have already completed the test, but you can always visit if you want to redo the test again, if you want to revise or if you want to improve the scores. After understanding the concept, you can always revisit it. A blue bubble shows that you are yet to take the test, you have not started with the test. A yellow bubble indicates that you have started the test but you have not completed. Sometimes it so happens you would have started the test, but after 4 or 5 questions you would have realized it, you are not really thorough with the concepts and you need to learn the concepts. So at that point of time you might leave the test and go, and it's an indicator, it's a reminder for you to come and complete the test. Now, if you look at the chapters, each chapter has varied number of tests, and this is created based on their importance in the NEET exam and also the complexity of the text. So once you have completed all the tests, you can take a red bubble test, that is randomized test, wherein questions will be asked of different difficulty levels, and it will also help you to analyze whether you have understood the concept and you are able to apply the concept. Now here if you click on revision notes, now I'm sure you're all eagerly waiting for your exams. Now you also must be wondering what are the different kinds of questions that might fare in NEET exams, especially in biology, because it carries half the weightage of your paper. And from our analysis, we have realized it that we have three kinds of questions. One is directly from NCERT. Approximately 68 to 70 questions in the examination are directly from NCERT books. They can be attempted easily by those who read NCERT meticulously. And direct from the previous question papers, if you look around 8 to 10 questions are asked straight from the past papers, either with modifications, slight modifications, or without any modification. And the third type will be the modified questions, which will be about 8 to 10 in number, and they become the game changer because they wouldn't have appeared anywhere in the test of NEET or AIPMT. It will be the first time you will be seeing those questions, though they are all NCERT based. Unless you are thorough with the concept and you know how to apply, attacking such questions requires a little practice, and that is what decides your score. Now I hope you have got the framework. Now today's class, I also hope you got to navigate through the website. In today's class, I will be revising the key concepts, do a practice test based on the concept learned, and last 5 minutes I shall answer your queries. Now let us get back to the NEET revision notes. So this is a NEET revision notes which has been prepared by the tutors. Or to learn for your NEET exams, and the entire 10 textbooks have been summarized into 135 pages for you to understand better, so that you know how to answer, revise before you go to the exams in a very precise manner and in a very comprehensive manner. Today we will be doing this lesson, Principles of Inheritance and Variation. Now in the beginning we would have given you all the important definitions which you are expected to learn. Now I'm going to start with what is inheritance and what is variation. Now genetics, you know, it's a study of heredity and variations. The term genetics was coined by Bateson in 1905. Genetics is generally of two types. One is the forward genetics. It involves initial study on the basis of phenotype, leading ultimately to the study of DNA sequencing comprising the gene for this phenotype. And reverse genetics, it involves initial study with a DNA segment, the phenotypic of which we do not know, and then introduce the segment in some plant or animal to study its phenotypic effect. Now, heredity is a transmission of characteristics from parents to offsprings. Such transmissible characters are called hereditary characters. Hereditary traits or characters are specified by genes, which are integral parts of the chromosomes. Now, variations are the differences in the morphological, physiological, cytological, and behavioral characters shown by the individuals of the same species, race, and family. The main source of variations are crossing over, which happens during the meiosis, chance combination of chromosomes during meiosis, fertilization, and mutation. And mutation is the ultimate source of variations. Mutations are sudden heritable changes. Now, let us look, before I'll explain all these terminologies based on Mendel's work. So before that, let us know about Mendelian principles and Mendel's work. Now, you know, Begar J. Mendel was an Austrian monk who performed breeding experiments on the Pisum sativum or garden pea, and he is credited to be the father of genetics. Mendel did not know about chromosomes or DNA, and he did not know anything about genes. By observing the number and types of phenotypes in the offsprings, Mendel developed the rules of inheritance, which were known as Mendel's law. Mendel used garden pea for his experiments because of its well-defined characters. The pea flowers were hermaphrodite, that is, they were bisexual. They were predominantly self-fertilizing, but it was also possible to cross-pollinate them very easily. And also because removal of anthers, that is emasculation, could be done very easily. And Mendel's success was mainly based on the fact that he considered a single character at one term. Now we have seen 7 traits he took it. He individually worked with these plants, and then he was able to tabulate his results, and then he gave a statistical interpretation for his experiments. Now let us look at the different characteristics what Mendel had selected. In case of stem height, he took a tall plant and a dwarf plant. In case of flower position, plants that had axial position were flowers, and flowers that were present at the terminal position were taken in. Pod color green versus yellow, pod shape full or constricted, flower color violet or white, seed color were yellow or green, and seed shape round and wrinkled. So whatever you see in the first column, they were all the dominant traits, and the second column were all the recessive traits. So this is the diagrammatic representation of monohybrid cross. So he selected a pure parental plants that were tall and dwarf. So to obtain this, he self-pollinated the tall plants continuously for 6 to 7 generations. And similarly, he self-pollinated the dwarf plants until he got the pure lines. Pure lines means these plants, whenever they are self-pollinated, should be true to their character. And this is what we call it as a pure line. And Mendel got a pure lines for as his parents for his experiments. Then he brought in about a hybridization. That means to say he removed the pollen from the dwarf plant and dusted it on the stigma, and then he saw what would, then he got a generation from this plant, which was called as the F1 generation. So when he did it, he got all tall plants in the F1 generation. And then he thought that there is one factor which is expressing itself in the F1 generation, which he called it as a dominant trait. In the F1 generation, he brought about self-pollination, and after self-pollination, he saw that in the F2 generation, there were 3 tall plants and 1 dwarf plant. That means to say the dwarf plant, which was hidden in the F1 generation, had come back in the F2 generation. And today we are able to interpret the results in this way. So this is a diagrammatic representation. If you look at capital T, and lowercase t, these are the parents. So this is the homozygous. So when both the alleles, when both the genes are similar, we call such a thing, such a pair as an homozygous alleles, and this is a homozygous allele. So you know that this will produce gametes, and these gametes will combine randomly. So this one, what you get as capital T, lowercase t, was a F1 generation. It was again a tall plant, and this condition is called as heterozygous tall or heterozygous status because there are 2 dissimilar alleles. Now, when these were self, so self means the mother will also have the same genotype father will also carry the male parent will also carry the same genes. So when he did it, he got capital T was also tall, capital T low case T was also tall, and small low case T and low case T was the dominant. So out of the 4 plants, 75% or 3 plants were tall and only one was dwarf. Now the external morphology, what you see of a plant, that is what is known as a phenotypic ratio or a phenotype, and the ratio obtained based on the phenotype is called the phenotypic ratio. So here you see the tall is to dwarf. So you are getting 3 tall plants is to 1 dwarf plant, and this is a phenotypic ratio. Now if I ask you what are the kinds of genes that make up these tall plants and the dwarf plants, that ratio analysis is called as genetic makeup of an organism is called as the genotypic ratio. Now, if you look this is capital T is homozygous tall, capital T lowercase t is heterozygous tall, and lowercase t is a dwarf. And this is what we call it as the genotypic ratio. Now, the trait that is expressed both in homozygous condition as well as in heterozygous condition is said to be a dominant trait, and the trait that is expressed only in the homozygous condition, we call it as a recessive trait. See here, capital T or capital T lowercase t corresponds to height. So this means it is a character tallness is dominant over dwarfness, and dwarfness is able to express only when there are similar genes, that is small t lowercase t and lowercase t, and that is why we call it this trait dwarf as a recessive trait. So I hope you have understood what is dominant and what is recessive, homozygous and heterozygous. Now, based on this, he proposed his law of dominance. It states that expression of only one of the forms of the parental traits is possible in the F1 hybrid. That in the F1 hybrid, the trait that is always displayed is described as the dominant trait, and the trait that disappears is known as the recessive trait. Disappears only in the F1 generation is known as the recessive trait. And based on this, he also proposed another law, that is law of segregation. It states that members of each pair of alleles of a gene separate when gametes are produced in meiosis. Mendel's law of segregation is universal, and it is without any exception, and it is also called law of purity of gametes or law of splitting of hybrids, because gametes always remain pure, and a gamete may carry either the dominant or the recessive factor, but never the both. Now the next concept you are supposed to know is the test cross. So if you look at a test cross, we are able to analyze that we are testing something. What is it we are going to test? Now you have got a plant and you want to see whether it is homozygous or heterozygous. So how do we do that? That testing is what is done by a test cross. So here what we are doing is unknown flower we are taking, a plant with an unknown flower which has produced a purple color. Now this we do not know whether it is homozygous or heterozygous dominant. So we are crossing it with a recessive parent, which is a plant with a white flower. So when we do the crossing, we are able to get all the flowers here are violet. That means to say it has both the genes, that is all the genes here are WW, that is the dominant trait is present here. If the dominant trait is present and the ratio is purple flowers to white flowers is 4:0, then our purple plant, the plant under consideration, is producing the flowers with violet flower, which are all homozygous purple. Now the same cross, if we are repeating it and we are getting 50% of purple flowers and 50% white flowers, then it becomes a heterozygous flower. That means the plant is got the characteristic of both homozygous, that is a dominant and a recessive gene, as a result of which we are able to see 50% of plants being white and 50% of the plants being purple. So this is about the test cross. Now there are deviations from the Mendel's traits, and these two I will explain now, because it is related. It is like a monohybrid cross. Now it is incomplete dominance. So we have seen dominance means a character is getting expressed both in homozygous condition as well as in heterozygous condition. Now when you cross two plants of snapdragon or evening primrose, one plant has got a red colored flower, the other one is producing a white colored flower. So when you cross them, you have to get either red or you have to get white. Then you will understand whether it is a dominant trait or a recessive trait. But here what has happened, you got an F1 generation all pink flowers. That means to say neither red was able to express itself nor white it was trying to express itself, but both were trying to express together. That means to say the color red was not completely dominant and so was white which was not completely dominant. Hence there was a mixture. So pink is lighter than red but it is darker than pure white. So this is what we call it as incomplete dominance, and this is how the ratios are being worked out. Here if you look at it, capital R, if the genotype is capital R, the plant will produce a red flower. If it is capital R small r, then the plant is going to produce a pink flower, and if it is lowercase r or lowercase r, then it is going to produce a white flower. So here the phenotypic ratio and genotypic ratio are the same, that is 1:2:1. So this ratio is similar, that is genotypic ratio and phenotypic ratio of 1:2:1 is similar to genotypic ratio of monohybrid cross. Now let us look at codominance. So as the name suggests, both are equal partners. So both of us are going to express, when both the genes try to express themselves and both the expressions are seen in the organism, then you call it as codominance. And a classical example would be the blood group inheritance. So we know that blood group is controlled by P genes, that is gene IA, IB and I. Now, if you look at the dominant factor, IA gene is dominant over O, IB gene is dominant over O, but IA and IB are codominant. So the organism will be having a blood group A if its genetic makeup is IA or IA. It will be having B if the genetic makeup is IB, that is homozygous, or IBIO heterozygous. O group if it has got I and I only, that is both are recessive. If it has got IA and IB, the blood group is going to be AB. Here what is happening? Very clearly A and B are codominant. Both are trying to express their character and both express, they are not masking the effect of each other, but both are getting expressed, and this is known as codominance. Now with this, let us get into dihybrid cross. So in a monohybrid cross, you saw a cross between two parents which differ with respect to one character. Now dihybrid cross is a cross wherein the parents differ with respect to two characters. So Mendel took a round seed with a yellow seed, round seed with a yellow color, wrinkled seed with green color. So round and yellow are dominant traits over wrinkled and green. So the parental generation were pure lines, and then you got a hybrid, which is a dihybrid, but that is also expressing round yellow. That means to say round and yellow were dominant traits. Now let us see how the dihybrid cross ratio goes. So if you look at the working here, you will be seeing at the end, round yellow is 9, round green is 3 in number, wrinkled yellow is 3 in number, and wrinkled green is 1 in number. That means to say round yellow and wrinkled green, the parental types are seen, that is 9:1. And then round green and wrinkled yellow are newly formed combinations and they are called as recombinants. So the genes are combining amongst themselves in independent manner, and there is an assortment of the genes. And this is what is the dihybrid phenotypic ratio, that is 9:3:1. Here 9 and 1 represent the original parental combination, 3:3 that is the recombinant ratio. So based on this, he proposed his law of independent assortment. It states that pairs of alleles separate independent of each other during gamete formation. So when a dihybrid cross is made and offsprings of F1 generation are self-bred, dihybrid ratio 9:3:1 is got. So this is what is all about the dihybrid cross ratio. Now you are seen. So this is a typical Mendelian distribution. Monohybrid cross is also a typical Mendelian inheritance. Now what you saw, incomplete dominance and codominance, are deviations from the Mendel. I covered it because we were doing it just and it becomes easier for you to understand. Now let us look at so linkage was proposed by Morgan. He was the first one who studied linkage in the Drosophila, but it is a tendency for the alleles of different genes to be passed together from one generation to next. If the chromosomes in a gamete producing cell are not rearranged or lack recombination during meiosis, it would be expected that all the genes on a particular chromosome would be transferred together into which gamete will get that particular chromosome. That is, the genes are linked together on their chromosome. Actually, linkage was first discovered by Bateson, Saunders, and Punnett when they were working with sweet pea. They found the test cross ratio to be 7:1:1:7 instead of 1:1:1:1 and described it as coupling and repulsion. Linkage is an exception to the law of independent assortment. And T.H. Morgan reported linkage in Drosophila, and he gave the term linkage to this phenomenon. He also reported that coupling and repulsion are two aspects of the linkage. A linkage group is linearly arranged group of genes. So if the genes are close together, then they will be able to they will be linked together because synapses crossing over will not take place. But when the genes are far apart, then the chances of recombinations are very high. So if they give you in the exam a question showing 3 or 4 genes and they will ask you which of the genes can show the linkage, you have to identify the 2 genes that are very close together and you have to say those 2 genes will be taken as a linkage group, and the genes that are farther apart, they will not be showing the linkage and they will be open for recombination. Now if you look at the linkage group, if you look at the number of linkage group will correspond to the haploid number of chromosomes. For example, if you take Drosophila, in Drosophila the haploid number is 4. That is N is equal to 4. The number of linkage groups will be 4. In garden tree, if you look at it, the linkage group will be the haploid number is 7. So linkage group will also be 7. In males, N is equal to 10. Linkage group will be 10. In Homo sapien, N is equal to 23. So the linkage group will also be 23. Now, what are the types of linkage? The linkages can be a complete linkage or it could be incomplete linkage. When only one parental combination are produced, then the linkage is called complete linkage. It is exhibited when the genes for a particular character are present very close to one another. It is rarely found in nature, but crossing over remains absent in this case, as in male Drosophila, female silkworm, etc. Incomplete linkage, when some recombinations are also produced along with the parental combination, then it is called as incomplete linkage. Now, for example, if you look at it in the test cross ratio when they were working with the sweet pollen, they found that is the purple color long pollen is what they were working about it. They should have got, if there were no recombinations, then they would have got the ratio as 8:8, but the 7:1:1:7 shows that it was an incomplete linkage. Now, the next concept we had to know is sex determination. So we know that in dioecious plants, there are several means to determine the sex. Chromosomes involved in sex determination are called sex chromosomes, or they are also called as allosomes. And the chromosomes that control the body traits are called as the autosomes. Those sex chromosomes provide the most common means of sex determination. It is not the only mechanism. In bees, if you look, males are haploid while females are diploid. Sex may be determined by a single allele or multiple alleles, as in some wasps, and sometimes environmental factors can also change the sex. That is, it is not genetic. That is what I'm trying to say. These are indeterminate genetic sex determinating mechanisms. For instance, the temperature at which the eggs are incubated will determine the type of the sex in the turtles. In some species, warm nests will eat males and cool nests will eat mostly females. And in some other species of turtles, it is reversed. Now, there are 4 basic types of chromosomal mechanism. The first, if we look at it, it is the humans XXXY system. So whichever system we talk, the first one what we use is for the female. That is why we are saying it as XXXY system, in which females are homomorphic. That is, females will produce the eggs all carrying only the X chromosomes, whereas males will produce sperms, half of them carrying X chromosome and half of them carrying the Y chromosome. So when an X-bearing sperm fertilizes the egg, it results in a female. When a Y-bearing sperm fertilizes the egg, it will result in a male organism. Similarly, in the Drosophila also, if you look at it, it is male XY and female XX. So male here it will produce half the sperms carrying X chromosome, half the Y, and then here you find it that the eggs are all homogametic, producing only eggs carrying X. And when the fertilization happens with X sperm, it results in a female Drosophila. When the fertilization occurs with the sperm with Y chromosome, then it turns out to be a male. Now, in birds, you have ZZ and ZW system, in which females are heteromorphic. ZW and males are homomorphic. That is, this occurs in birds, fishes, and moths. It is essentially the opposite of XY in mammals. So here you look at it, the male has got ZZ, so it is homogametic, and female is the ZW, so it has got the both the kinds of eggs. Some eggs will produce the Z sperms and some eggs will have W sperms. When the Z sperm fertilizes the Z egg fertilizes the sperm, then it will result in a ZZ combination, resulting in a male. And if an egg having the W chromosome fertilizes the sperm, then it will result in a female. Now, is it only the pattern? No. So if you look at genic balance theory, sex determination in the Drosophila, there are different types. So here, this is what we call it as the genic balance theory, which states that X chromosome determines the sex of the individual, and that sex is a dosage phenomenon where the ratio of the amount of X relative to the autosomes determine the sex. In addition, environmental effects can also influence the development of intersex. Now, genic balance theory was originally proposed by Calvin Bridges in 1921. Gynandromorph is a dioecious organism with sexual mosaic. Gynandromorph is typically male in certain portions of the body and typically female in others. It means body of gynandromorph will be half male and half female, and this term was coined by Goldschmidt. Gynandromorphism has been reported in Drosophila, human, silkworms, bees, butterflies, beetles, etc. Now, if you look at here, let me explain. Normal female, the number of chromosomes will be XX plus 2n. So if you take X by autosome, that is 2 by 2, the ratio is 1. In normal male, it is 2 by 1, so it is, 1 by 2, it is going to be 0.5. In meta female, we also call it a super female. There will be 3 X chromosomes along with the normal autosomes. So that will result in 1.5 as the ratio. In meta male, there is only going to be 1 X chromosome. So it is going to be 1 by 3. So it is 0.33 is the ratio. In intersex will be 2 X chromosomes, but autosomes will be 3 in number. So there is a reverse, and that is why it results in a gynandromorph or a intersexed organism with a ratio of 0.67. So if they give you a question asking you this is the chromosomal complement, and to identify with the choices as normal, male, female, or whatever it is, you take the number of X chromosomes and divided by the autosome, and what depending upon the ratio. If the ratio is 1, it is normal female. If it is a half or 0.5, it is normal male. If it is 1.5, it is meta female. If it is 0.33, that is less than 0.5, then it is a meta male, and intersex, it is between greater than 0.5 but less than 1, it is an intersex. So like this, you should be able to solve the problem if it is asked in the exam. Now let us look at the inheritance patterns. So what are the inheritance patterns we have seen? One is sex-linked inheritance and other is autosome-linked inheritance. So sex-linked inheritance can be sex-linked dominance inheritance and sex-linked recessive inheritance. Similarly, autosome-linked inheritance and autosomal recessive inheritance. Now, if we look at the characters or traits which are carried, the inheritance is based on the characters that are linked to the sex chromosomes, then you call it as the sex-linked inheritance. Again, T.H. Morgan observed sex-linked inheritance in Drosophila. In humans, if you look at it, color blindness and hemophilia are important sex-linked diseases. The genes located are exclusively on the X chromosomes and are called X-linked, and the genes that occur only on Y chromosomes are called polymeric genes, whereas the genes present in the homologous region in the sex chromosomes are called XY-linked genes. Now, if you look at the status, so if it is sex-linked dominance, if the trait is shown as a dominant trait and it is getting inherited, we call it as sex-linked dominant trait. If it is expressed in the recessive status only, then you call it as sex-linked recessive. Similarly, we have autosomal dominant inheritance. If the trait is rare, most meetings are between heterozygotes, that is, who are affected, and homozygous unaffected recessives, which lead predominantly to 1:1 phenotypic ratios in all the children, regardless of the sex. Autosomal recessive inheritance, generations are often but not always skipped. There should be equal distribution among the sexes. If both the parents are affected, all the children will be affected. This is often found in consanguineous marriages, and most affected children will have normal parents. So, what happens in the consanguineous marriages, where the marriage is between the close relatives, then the possibilities of genetic disorders are very high, because in the parents they might appear normal, because if the trait is in the recessive status, parents will be obviously normal, but they will be the carriers. And in the children, if both the recessive genes come together, the character gets expressed. Now let us look at some of the genetic disorders. Okay. So these are the Mendelian disorders, means which follows the Mendelian character. So this is what are these are the diseases, and that's why we call it as the Mendelian disorder. The inheritance pattern will be similar to Mendelian inheritance. Example would be hemophilia, color blindness, sickle cell anemia, phenylketonuria, and thalassemia. Now let me briefly explain to you, but you should know all these diseases because it's a very important question. One question will be sure from these diseases. Hemophilia is X-linked recessive disease, which was first studied by John Porto in 1803. Hemophilia was common in royal families of Europe. Alden discovered pedigree of hemophilia in royal family, which started from Queen Victoria due to mutation in their gametes. Hemophilia A is characterized by lack of anti-hemophilic globulin, which is also known as factor VIII. About 4 to 5 cases of hemophilia belong to hemophilia A. Hemophilia B or Christmas disease results from a defect in plasma thromboplastic component or factor IX. I have already told you that during blood clotting, there are 13 factors that are responsible. Only if all the 13 factors are intact, clotting mechanism will be intact. If there is any disruption in the factors, because it is all a cascade of events, then it will hamper the clotting in the organism. As hemophilia is caused by a recessive X-linked gene, a woman may carry the disease and is called as the carrier because it is present on the X chromosome. So if a woman has got two sets of X chromosomes, one is defective and one is normal, she will only be the carrier and she will not suffer from the disease. But males will be having only one X chromosome, and if they get a defective gene, they become hemophilic by nature. Now, color blindness is the condition in which certain colors can't be distinguished due to lack of one or more color absorbing pigments in the cone cells of the retina. In humans, the most common color blindness is red-green color blindness, which is a sex-linked defect caused by a recessive gene and is more common in males than in females. Now, the next one will be sickle cell anemia. It's an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the parents are carriers for the gene. The disease is controlled by a single pair of allele, HBA and HBS. Out of the three possible genotypes, only homozygous individuals for HBS will show the diseased phenotype. Heterozygous individuals will appear apparently unaffected, but they are the carriers of the disease, as there are 50% probability of transmission of the mutant gene to the progeny that's exhibiting sickle cell trait. The mutant hemoglobin molecule will undergo polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle cell structure. The next disease would be phenylketonuria. Again, it is an autosomal disease. So, hemophilia and color blindness, the genes are located on the sex chromosome. But in sickle cell anemia, phenylketonuria, and thalassemia, it is related to the autosomes or the somatic chromosomes. This is also an autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. This phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. These are excreted to urine because of its poor absorption by kidney. And thalassemia is an inherited autosomal codominant blood disease. In thalassemia, the genetic defect results in reduced rate of synthesis of one of the globin chains that make up hemoglobin. Reduced synthesis of one of the globin chains can cause the formation of abnormal hemoglobin molecules, thus causing anemia. Now let us look at the chromosomal disorders. You had Down syndrome, Klinefelter syndrome, and Turner syndrome. In the Down syndrome, this is the presence of an additional copy of chromosome number 21, which we also call it as trisomy of 21. The affected individual will be short stature with small round head, furrowed tongue, and partially open mouth. Now, Klinefelter syndrome, this genetic disorder is due to the presence of an additional copy of X chromosome, resulting in the karyotype of 47, that is XXY. So, it will be 22 plus XXY. Some such individuals will have overall masculine development. However, they also exhibit feminine development like gynecomastia, and such individuals are sterile. Now, Turner syndrome, this disorder is due to the absence of one of the X chromosomes, that is 45 with XO, and these females are sterile as ovaries are rudimentary, besides other features including lack of other secondary sexual characters. Sorry, the last thing I had to explain to you is about pedigree analysis. So pedigree analysis is very, very important from a genetical standpoint of view. Here, the analysis of traits in several generations of a family, when you analyze, you call it as a pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generation. And if there are any defects that are there, then by analyzing the pedigree, they will be able to identify to tell what kind of problems will be occurring in the unborn kids. And so that parents can always take up a decision to identify whether they had to go with that or abort the fetus or not to have the children. See, some of the genetical disorders can also be found by amniocentesis. From that, they will also be able to analyze if the child is suffering from any genetic disorder. Based on the pedigree analysis and also the reports of amniocentesis, a couple can decide whether to go with the pregnancy or abort it or medically terminate the pregnancy. Now, in a pedigree analysis, this will be the kind of representations you make. A male is represented by a box, female by a circle. If you do not know the sex, it is represented by a rhombus. If it is shaded, then the individuals are affected. If both a male and female are joined by one line, that means they have got into marriage and they are unrelated. If both, there is a marriage between consanguineous marrying or close relatives, you will be showing it by a double line, and this will be the parental generation. You draw a vertical line and then horizontal line to show the males or females. This will become the first generation, and that is the kids of this generation. And now if you look at it, parents who are normal have got a child which is a male which is affected, and if the number is written within a box with unspecified sex, that means to say 5 kids are born, or it could be 3 but their sexes are unknown in the analysis. So with this, we complete all the concepts that are very, very important from NEET point of view. Here in this, whatever I have explained have been put in a nutshell in 1 or 2 to 3 pages, so that it is easier for you to understand, and it also helps you to get the meaning of the concept ready, and it also helps you to identify the definition. Sometimes definitions can be asked where you might find it very difficult to answer because your NCERT textbook does not give you the crisp definitions. Now let us come to do a practice test. here. Okay, the child looks similar to the parents, but not the same is due to, it is similar due to inheritance, but not the same due to variations. So it is inheritance and variation. Indian breed of cow found in Punjab is Sahibal. The dwarf plants is to tall plants in the F2 generation. See, look at the question. It is not tall is to dwarf. It is dwarf is to tall. So it is going to be 1 is to 3. Dash are the units of inheritance. Genes are the units of inheritance. When the same gene codes for opposite traits, we call them as alleles. See, earlier they used to call it, even if the opposite trait, call it as alleles, but now they have modified their definition. When a pair is there, whether it is homozygous or heterozygous, you call them as alleles. Okay. The genotype capital T lowercase t codes for dash phenotype, tall phenotype. Even though the genotype capital T lowercase t carries an allele for tall and dwarf trait, the plant displays a tall phenotype, and this phenomenon is called dominance. The character that is genotypically expressed but not evident phenotypically is called recessive. The cross between capital T and small lowercase t is, so it's a monohybrid cross. While you look at the other part of the question, it is a dihybrid cross. So it is monohybrid and other is a dihybrid. The pictorial representation of Mendel's law was done by Reginald C. Punnett. That is what we call it as a Punnett square or a checkerboard. The crossing of a dominant phenotype with a recessive parent is called a, so we have seen that is a test cross. So that's why we do not know whether the dominant phenotype is homozygous or heterozygous. So by crossing it with a recessive parent, if we get 4 is to 0, then it is a dominant trait. If we get 1 is to 1 ratio, then it is a heterozygous trait, dominant. So that is why we do a test cross. According to law of dominance, it's not law, it's law of dominance, factors occurs in pairs. The phenotypic proportion of 3 is to 1 is explained by law of segregation. Heterozygous ones produce two kinds of gametes, each having one allele with equal proportions, can be explained by, just a minute. Phenotypic proportion of 3 is to 1 is explained by law of dominance. I'm sorry. So this is going to be law of segregation. Stable discrete units that control the expression of traits is called genes. So I'm ending the test. Let us see what the answers are. So all the answers are correct. If any question, any answer is wrong, then you will be seeing the red colored box. If you just click on that box, your answer and the correct answer along with the explanation will be given. If you have not attempted, it will be showing in a yellow color. Again, you click on that box, you will be able to see the correct answer along with the explanation. Since you are giving an entrance exam and it has got negative correction, I tell you that I suggest you don't attempt the questions if you are not sure of the answer, and don't do to try like inky pinky ponky to get the correct answer, because it's going to cost you -1 if it is wrong from your total marks. It's not out of 720, but whatever the correct answers you have given, that will give you into 4 minus your incorrect ones. So you have to be very careful when you do this. So today we are coming to an end of today's class. In the NEET series, you also have NEET full test. In this test, if you see, we have created around 38 tests, out of which 15 tests are created by the tutors, taking into consideration the complexities and the syllabus of the NEET and looking at the type of questions. and they are all tutor prepared tests, and other 20 odd papers are of past years papers. So if you are able to solve all these things, you will be able to do NEET at ease. You will not have any issues. So I would suggest or recommend you to go in for this package. If you still want to ask anything, you can use our ask feature and post your queries. And if you want one-to-one tutoring for any particular lesson or for any particular subject, you can always get in touch with the tutors. And if you have any other query, you can mail to info@clay6 to get your queries resolved. Thank you, children.